Welcome to the first part of our podcast on the cardiovascular system. In this podcast, we're going to be examining the early events that form the primitive heart, and then we're also going to be looking at the early influences of the different cell types on this heart, as well as the early embryonic circulation. So, cardiovascular system, the first system to be functional in the embryo, and we're really referring to the heart in this part. It appears in the mid-third week, and it's going to be functional in the fourth. Now, if you recall, we principally talk about structures forming in the fourth week and functioning sometimes later, but this one starts to form a little bit earlier and is fully functional by that fourth week of development. Now, the heart, while we see it as a singular structure, it certainly is going to be complex, and it's devised from a couple of different mesodermal types, as well as from an ectodermal type. We can see our splanchnic mesoderm, and this is going to be our cardiac primordium, and this is what is going to be at that anterior part by the septum transversum. We also have paraxial and lateral plate mesoderm that are going to contribute as well. And then we have migrating neural crests that come in and help to partition and form the outflow regions of the heart. And we'll get to those specifics as we get to the outflow of the heart and its development. But as we begin, we have our early precardiac mesoderm, again, the septum transversum region, and then we're also going to have lateral around the developing embryo itself. Now, this is at the time that we still have our trilaminar disc. We've completed neurulation. And so in our lateral mesoderm, we are going to begin to form blood islands. You're going to have de novo development from mesenchymal cells into hollowed out vessels, and in the center of these, you're actually going to form some blood cells, in fact. And some of these coalesce in the head region on the lateral aspects, and we refer to these strands as angioblastic cords. Now, angiogenesis as a term means you are going to form new blood vessels from existing vessels. The term vasculogenesis is what we really refer to in that early formation of the blood islands, where you develop blood cells from precursor mesenchymal tissue. Now, we're going to see that we have these paired cords, right and left. And so these are going to form early endocardial heart tubes. We're first going to be solid. Then we're going to undergo apoptosis to form the lateral endocardial heart tubes. Notice that's still plural. But then as the lateral folds of the embryo begin to curve around, that's where we are going to fuse these lateral tubes along the midline of the embryo. The head fold is bringing down that precardiac mesoderm from the septum transversum region. That's going to coalesce together into our single tubular heart that's going to be now along the median plane of the body. So here in our illustrations, we can see, looking in an overhead view, if we were in the amnion looking down, here we can see our neurulated embryonic disc, ectoderm on the surface. Here is our septum transversum, but our precardiac mesoderm. We're also going to have our lateral mesoderm on each side. And as we cut through a cross-sectional view, we can see our solid angioblastic cords. Again, this is going to be our pericardio-peritoneal canals, and we've got that on either side. Now, if we have a mid-sagittal cut and look down, here we can see septum transversum, and then our cardiac mesoderm, and within the same region, we have our lateral angioblastic cords. Now, if we continue looking at this perspective, again, we're still looking downward, and as we look at this cross-sectional view, you can see now we have our endocardial heart tubes, these lateral tubes, but now the amnion is pushing those lateral folds closer and closer together. Here we have our developing foregut. A little bit closer, we're beginning to fuse our mesoderm together. Now we're going to begin to fuse these endocardial heart tubes together into our single tubular heart. We're still attached with our dorsal mesoderm. In this case, we call this the dorsal mesocardium. And a little bit further development, we're going to lose that dorsal mesocardial attachment, and this is where we have our single tubular heart comprised of the inner lining, the endocardium, and our myocardial later. And then lastly, we will add, subsequent to this, from migration from outside the heart, our third layer on top, which will be the epicardium. Now let's watch a video that's going to put this into motion to show the fusion of these angioblastic cords into our single tubular heart. During the time that we're forming the heart, we're also going to be forming our major embryonic veins. These are going to come from that vasculogenesis and the blood islands coalescing together, also from the folding of the embryo. So the major veins that we have in embryonic development are going to be the vitelline veins. These are largely depleted of oxygen. They're coming back from the yolk sac. We have umbilical veins. These are coming back from the placenta, so they are going to be rich in oxygen. They're veins, remember, so returning to the heart is the reason we call them veins, regardless of the oxygen content. But then we also have cardinal veins. These are oxygen-depleted blood returning back to the heart from the embryo. Now, these cardinal veins are going to be remodeled during development. We're not going to spend a lot of time talking about that remodeling that occurs, but it is extensive in the change that happens from embryonic to human vascular supply. So in our illustration, we can see our heart situated in the middle, and it's somewhat upside down from the way that we envision it. The atrium is actually going to be more caudal, and at this point we have a single atrium, a single ventricle. We have an outflow tract, which is going to be here, and an atrioventricular canal situated between the atrium and ventricle. Sometimes this is referred to as the aortic sac, but more commonly in developmental cardiology, we call this the outflow tract. Here we have our yolk sac. So we've got our vitelline veins bringing blood back to the heart. Here we have our chorionic villi, and so this is our umbilical cord. We have our umbilical artery and umbilical vein. And in this case, I wish they had changed the colors, because this is not blue. This is actually red, because we have oxygen-rich blood, and this is going to be largely oxygen-depleted in the artery. And then lastly, we have our cardinal veins that are coming in, the anterior, the common, and the posterior cardinal veins. This is the anterior cardinal. Here's the posterior cardinal, and the common cardinal is the union that we see here. So the vitelline veins, as we mentioned, are returning blood back from the yolk sac, and they're going to empty blood into the sinus venosus, and we'll talk about that as we get more specific into heart development. Now, the vitelline veins don't completely degenerate. They're going to be recycled and remodeled The adult derivatives will be the hepatic veins, and we're going to form those from the right vitelline vein, and the portal, the hepatic portal vein. And we're going to use portions of both the right and left vitelline veins to do that. The umbilical veins, as we've seen, this is going to be bringing back from the placenta. And we're going to see this also empties into the sinus venosus, which is a common collection point then before blood is emptied into the atria. We're going to see that the right umbilical vein is going to degenerate near the end of our embryonic period. However, the left umbilical vein is going to persist. It's going to form the single umbilical vein, and it's also going to form part of the ductus venosus. Now, we're going to see this ductus venosus is going to be important. We have a couple of bypasses that happen. We bypass the liver, we bypass the lungs, and this bypass of the liver is going to be our ductus venosus. And when we explore the totality of the circulation, we'll see that bypass. We don't need the blood to be detoxed because that's what the placenta does. We don't need the lungs for gas exchange because that's what the placenta does. Hence, for embryonic and fetal circulation, these are some of the major differences we're going to see between the fetus and the neonatal vascular supply and circulation. Now, the cardinal veins, again, this is going to be the major venous drainage. You can think of these similar to the superior vena cava and the inferior vena cava that we have in the adult body. And we'll see some of those are, in fact, derived from these. The anterior and posterior are going to drain those portions of the embryo respectively. And again, they are eventually going to dump into the sinus venosus, but they have a common union, which is called the common cardinal vein. And if you've taken comparative anatomy, you're familiar with these terms already. And then what we see from the cardinal veins, the superior vena cava, this is going to form from the right anterior cardinal vein, and our common iliac veins are going to form from our posterior cardinal veins. Now, here's our illustration just showing we've got our anterior cardinal vein on both sides. We've degenerating right umbilical vein. We're losing our left umbilical and vitelline veins. And here we have this ductus venosus, this bypass. Normally, we have our vascular supply coming in. We have our hepatic portal vein coming in. We have the hepatic veins that are draining the liver into our inferior vena cava. But in this case, here is our left umbilical vein. And instead of moving through the liver, we are going to bypass the liver to go from the umbilical vein through the ductus venosus right into the inferior vena cava and to the sinus venosus. That is our bypass. Now, for our arteries, we're going to see that we do have a dorsal aorta. This is going to supply the yolk sac, lantois, chorion. We have vitelline arteries. These are going to go to the yolk sac. And again, they're going to be drained by the vitelline veins. The vitelline arteries are going to become in the adult the celiac artery as well as the superior and inferior mesenteric arteries. Then we have the umbilical arteries. We have one umbilical vein, but typically two umbilical arteries. These are going to become the internal iliac arteries. This is the most proximal portion. And the distal portion are going to contribute to the medial umbilical ligament. Now, let's take just a bit of time before we move into the layers of our heart. Let's just watch a short video that explains the changes that are going to happen with our circulation. As we've seen in illustrations already, when we talk about our early tubular heart that is now medial, what we're going to see is the different layers we have. Certainly, we recognize the thickest, largest muscular layer, which is going to be our myocardium. We have mentioned our epithelial lining of the heart, which is basically identical to the endothelium of our blood cells, but since it is in the heart, we refer to it as the endocardium. Now, in these illustrations, we also see that there is an acellular, extracellular matrix that is situated between the myocardium and the endocardium. It's said to be gelatinous because it is very hydrated because of the composition of carbohydrates, sulfated molecules. They're very charged, and so this hydrated semi-solid space is simply referred to as cardiac jelly, and this is going to be important for allowing inductive molecules to cross that space. And as well, it's going to allow a substrate for cells to migrate. We're going to form mesenchymal cells that are going to be important to migrate, fill this area for the structural components of the heart. Now, as we've mentioned, the epicardium is going to be the last layer to form. And this is going to form from mesothelial or mesodermal cells that are going to be coming from the area of the sinus venosus. This area is now known as the proepicardium or the proepicardial organ. And these structures are going to contact the myocardial surface of the heart, and these endocardial precursor cells are going to begin to proliferate, and they're going to cover and invest the surface of the heart. And that's where we're going to form this outer, sometimes called the serous pericardium or the visceral pericardium, just the outer layer of the heart. So here in our illustration, we can see the myocardial layer on the outside now, the sort of spongy looking, as this artist tried to draw it, cardiac jelly region, and then the endocardium on the inside. This is a scanning electron micrograph view of an early embryonic heart. We have a single ventricle, this large bulbous region. This is our outflow tract. We have a single atrium, and then this is a cut portion of the collapsed, somewhat bag-like structure that's the sinus venosus, and we have the atrioventricular region somewhat behind and deep. What we're seeing in this area, we are seeing these several processes, cell-coded processes, and this is the proepicardial organ, or simply the proepicardium, and where it contacts the heart, we're going to see these cells actually, as they proliferate, they will move. And you can think of it as proliferation. Certainly they're going to be dividing and doing that, but they're also moving. They're migrating across the surface. It's not just mitotic activity pushing these cells across. You can see the difference between these rounded myocardial cells and these larger, more flattened epicardial cells as they begin to move. Here we can see that line of that investment where here we have epicardial cells and here we have bare myocardial cells. Close-up view of that junction, myocardium in the lower right, epicardium in the upper left. You can see all of these cell processes extending away and ruffling of the edges of these epicardial cells. These are indicative of cells that are actively migrating across the surface. So we're continuing to grow the heart. Now the heart's going to bend and fold, and part of this is because the embryo is growing. Part of it is because the anchorage points that we have within the embryo where the heart is attached. But because of that, we are going to divide the heart up into these distinct regions. Now, this is sort of starting backwards, and so I'm going to start at the bottom. Blood returning to the heart is going to first enter the sinus venosus. From the sinus venosus, blood will move into the single primitive atrium. Now, both atria are largely going to be derived from this precursor tissue. From the atrium, we'll move through an atrioventricular canal to a single ventricle. Now, this is going to form the future left ventricle. Because the outflow tract, we now divide into two structures. The most proximal part, and it's rather enlarged by this stage, we refer to as the bulbous cordus. And this is going to be the future right ventricle. And then the distal portion of the outflow tract we call the truncus arteriosus. So here we have our illustration, sinus venosus at the bottom, our common cardinal veins coming back in, primitive atrium, AV region, ventricle, bulbous cordus, truncus cordus, and then we get into our aortic arches or pharyngeal arch blood vessels. Now again, because of those anchorage points and because of the differential growth of the embryo versus that of the heart is going to not have enough room to grow between these anchorage points, and it is going to actually protrude out. And we refer to this as a U-shaped bend or a U-shaped heart. First we have a tube, then we're going to have a U-shape, later we'll have an S-shape. And because of all this bending, we are going to form different regions of the heart. Some are going to dilate, some are going to constrict, and this is also going to help us form the different compartments of the heart. Now, as we do this U-shaped looping, what's going to happen is now the atrium that was caudal, as well as the sinus venosus, this is going to move to a more dorsal position, and it's going to continue to move until we know the atrium is actually in a superior position relative to the ventricle. Now, we saw in an illustration, and we'll see it coming up again. At this point, our heart, even with the U-shaped bending, we're going to see that it is still suspended within our pericardial cavity by the dorsal mesentery. And since it's attached to the heart, we refer to this particular position as the dorsal mesocardium. And it was thought for a long time that the proepicardial organ was a part of this dorsal mesocardium, but it is pretty well established now that these are two completely separate structures. So once we put this together, if we had to trace blood from the placenta through the heart back to the placenta, what we would see is we have the embryo placenta exchanging materials. We're going to have our umbilical vein, and we don't have that written in there, but the umbilical vein coming to the sinus venosus. We have our sinoatrial valves, and we'll talk about those a little bit more. They're going to be really important, as well as the atrioventricular valves. But then we have our single atrium, the atrioventricular canal, the single ventricle, bulbous cordus, truncus arteriosus, aortic arches, dorsal aorta, and once again we have our umbilical artery going to the placenta, and that would conclude our circulation of the blood from the placenta back to the placenta. Now, as we look at this is more or less a coronal view of the heart. Here we have the sinus venosus with our cardinal veins coming in, atrium, atrioventricular canal, and we're going to spend quite a bit of time talking about these endocardial cushions here that are going to function as primitive unidirectional valves, but also are really important for a structural connectivity. Then we have our primordial ventricle that is going to be our left ventricle, bulbous cordus, which is going to be the precursor tissue for the right ventricle, and now we have our outflow tract. Now, spend a little bit of time talking about this atrioventricular region. We saw that picture, and it's important to understand that these structures, while they're functioning as primitive valves, they are also forming this very important structural bridge that is going to divide the atrioventricular canal into right and left halves. Also, it's going to be within this region where the endocardial cells are going to be induced to undergo what's referred to as an epithelial-mesenchymal transition. They're going to form mesenchymal cells, migrating cells that are going to move through that cardiac jelly, populate that region, and these cells are going to begin to lay down the collagenous extracellular matrix that's going to form the connective tissue for the valves and the septa of the heart. So here is a term that is often used by developmental biologists, and that these mesenchymal cells are the onlogon, which is a term which means the precursors are the cells that come before. They're the onlogon for the connective tissue of the valves and the septa. So here we can see our endocardial cushions. They are prescribed as dorsal and ventricle. And in these two-dimensional illustrations, it's a little challenging to see. But they're going to continue to enlarge and constrict that atrioventricular canal, but it's a bridge of tissue instead of just completely occluding the passageway. Because as you fuse these together, you can see by this arrow, blood is going to be able to circulate on the back side, or blood could pass on this foreground side. So you've got your right and your left atrioventricular canals, which is going to translate into our right or left flow and our tricuspid and bicuspid valves. Now, if we take another plane of section through and look at that cut view, now this is more familiar with what we're looking at. Instead of looking at a side view of the fused cushions, now we're looking at a dead on, if this were coming straight at you. And this is that solid column of tissue, and now we have divided the right versus the left atrioventricular canal. And I think you can also envision this is where we're going to form our valve leaflets and those cusps from the lateral portion of the AV region as well as from this fused portion of our endocardial cushions. So they play a huge role in the formation of the valves, but this is also going to be sort of a central columnar connecting type tissue for our atrial septa that is going to form to partition the right and left atria apart, and our ventricular septa that's going to grow up. If this were not here, you would have a common union and a mixing of the blood, and that would be a pretty severe congenital birth defect. And we'll talk about that toward the end of our third podcast. This is a immunofluorescence image showing a atrioventricular region. We have the myocardium here labeled by M, and you can see that in red. The E is the endocardial surface. This would be the lumen of the atrioventricular canal. And with two different antibodies have been used here. The green is an antibody against fibronectin, which is an adhesive glycoprotein used by migrating cells. And we can see that there are really no mesenchymal cells present in where we see the green. However, all of these black dots represent these migrating mesenchymal cells that form during the epithelial-mesenchymal transition at the endocardium, and they begin to migrate through this area. Now, the red that you see, and when you mix red and green together, you get this yellow color, so both are present, is a proteoglycan, a chondroitin sulfate proteoglycan that is produced by these migrating cells. And so this proteoglycan, and the antibody recognizes it, shows the leading edge of the migration of these mesenchymal cells. Here you can see the cell out in front completely surrounded by the staining for that proteoglycan. And so as these cells migrate, they're modifying the extracellular matrix, contributing to the formation of the connective tissue elements. And that's a nice image to show that these cells are migrating, these cells are conditioning, and they don't all pile up against the myocardium. They stay scattered throughout this cardiac jelly in this endocardial cushion tissue to form those connective tissue elements to support the formation of the valves, to anchor the septa. And without that very strong structural integrity of collagen, you would have severe birth defects.